Hi everyone, my name is Nishchal Kumar and it's January 21st, 2020, and I'm here to talk about cases 2 and 3 in the case connections assignment. So if we look at case 2, what we're looking at is essentially glucose uptake in tumor cells versus surrounding normal cells, and we are given an image of an MRI and a PET scan as well, or a PET scan. And the first question we were asked was, in normal tissue, how glucose uptake is regulated in the liver, the skeletal muscle, the brain, and in the adipose tissue or fat. So what I said was, since glucose can be taken up and permeate cell membranes, glucose transporters are required. These are named GLUT. Glucose is a hydrophilic molecule, and these transporters help glucose move down their concentration gradients. Glucose uptake is regulated by GLUT2 in the liver, which is always on the cell membrane regardless of the presence of insulin. The liver releases glucose to the bloodstream through processes like glycogenolysis and gluconeogenesis, and these two processes become the primary source of glucose for the brain and red blood cells as well. So similarly, skeletal muscle and adipose tissue both use another one of these glucose transporters named GLUT4, and they use it for localization to transport glucose to the plasma membrane. Since this process is insulin dependent, it is contained within cells in vesicles until insulin becomes present. So, when insulin does become present, it can bind to the correct receptor, and skeletal muscle begins the translocation of GLUT4 to the plasma membrane. This then allows for the liver and muscle to take up glucose. Now, lastly, in terms of the brain, GLUT1 and GLUT3 are the main regulators of glucose uptake. Unlike the previous process, glucose uptake in the brain is actually insulin independent. So GLUT1 expression is seen in the blood-brain barrier and red blood cells, while GLUT3 expression is mostly seen in the neurons in the brain. As we've learned before, the presence of GLUT3 is because the brain has a high affinity for glucose because of that low Km value. So this transport is crucial for the blood-brain barrier and helps in moving D-glucose to the brain. So those are the four ways in which glucose uptake is regulated in the liver, skeletal muscle, brain, and adipose tissue. So the next question we're asked is, how does glucose uptake in normal tissue compared to that of glucose uptake in the tumor cells? And why is that advantageous to the invasive tumor? So if we look at the two scans and compare them, we can see a clear distinction of the area where the tumor is. This unit has taught us that the brighter areas on the PET scan reveal the cell's elevated metabolism level. Upon seeing the scan, I believe that after D-glucose administration, there's a higher uptake of glucose within the tumor cells than the normal cells surrounding it. Glucose is taken up at a much greater rate in tumor cells as compared to normal cells, and according to some research I found, this difference is best explained by the Wartburg effect. In tumor cells, the cells take in glucose differently through fermentation to produce lactate, instead of resorting to mitochondrial oxidative phosphorylation. So as a result, more ATP is generated and proliferation of tumor cells are increased. This process would be advantageous for the invasive tumor cells because they can now undergo the glycolytic process more often and aerobically, which can lead to further proliferation. So the last question we're being asked is what modifications may have occurred in cancer cells to alter this kind of glucose uptake. And I read some research, and I believe that GLUT1 upregulates glucose transport in glioblastomas. And that's significant because it's vital, again, to the blood-brain barrier, like I said. So that is case 2. Now, if we move on to case 3, let's see. So what we were asked in case 3 was a 5-year-old patient who was a boy, was brought in by his mother because he appears sickly, drowsy, he's urinating frequently, he's always thirsty, and some of his blood levels were taken, and his insulin levels were taken as well and compared to a control group. And we're first asked what the table is telling us about the biochemical basis of what's going on with the patient. So what I said is, according to that table, the patient's blood insulin levels are significantly lower than the control. I mean, it's 0.8 to 6, so way lower than the control group. And even in an hour after receiving that glucose levels, his insulin levels do get higher, but they're still significantly lower than that control group. Again, it's 5 to 40. So again, much lower. So what I said is the patient has diabetes, and that can be shown in his symptoms like the persistent thirst, frequently urinating, drowsiness, and since the patient is young with a new onset of diabetes, this is most likely type 1. So now it's asking what would I predict the patient's fasted blood glucose levels to be given his current condition. And the options were above normal range, within the normal range, and below the normal range. And so I went on by explaining that for this patient, the beta cells in his pancreas that control insulin synthesis and release are being killed by his own immune system. And meanwhile, his alpha cells that generate glucagon cannot communicate effectively. So in turn, there is decreased insulin signaling and a subsequent increase in his blood glucose levels. This glucose buildup in the blood is because glucose cannot properly enter the cells and just stay in the bloodstream as a result. I would predict the patient's fasted glucose levels are probably above his normal range, the normal range, and the patient is not efficient in storing and using glucose and cannot produce insulin very well. So the last question I was asked was to list 2 regulatory enzymes that are likely to be active in this individual's current condition. And so the 2 enzymes that I picked that I believe were active in this patient's current condition was glucokinase and phosphofructokinase. So let me explain. So both hexokinase and glucokinase, as well as phosphofructokinase, are active in this patient's current condition. Glucokinase is responsible for converting glucose to glucose 6-phosphate. That's something we learned, about glycolysis very early in our studies here. And since the child is hyperglycemic and the process is happening within the beta cells of the pancreas and the liver, glucokinase is definitely present. Hexokinase is present in most tissues, but since this process is happening in the pancreas and liver, I'm going to say glucokinase is present. Even though hexokinase is present in most tissues. In the liver, glucokinase can only phosphorylate when glucose concentrations are high. And since we know that glucose levels are high in this patient, we can surmise that glucokinase is active. Phosphofructokinase converts fructose 6-phosphate to fructose 1,6-bisphosphate. According to some research I did, there's a high abundance of phosphofructokinase and glucokinase in patients that have type 1 diabetes. Thank you for listening, and those are the two case connections.